Okay, hi everyone. Thank you so much for joining. It is my pleasure to introduce Professor Wengong Jin for our early career seminar series today. Wengong is an assistant professor at the Khoury College of Computer Sciences at Northeastern University and a visiting research scientist in the Eric and Wendy Schmidt Center at the Broad Institute. He received his PhD from MIT CSAIL, where he was advised by professors Regina Barzilay and Tommi Jaakkola. His research has been recognized with the BroadIgnite Award, the Dimitris N Chorafas Prize, and the MIT EECS Outstanding Thesis Award. Professor Jin's work focuses on geometric and generative AI models for drug discovery and biology. During his PhD, he led the development of JT-VAE, one of the first deep generative models for molecular graphs, and he co-developed ChemProp, a widely used method for molecular property prediction. He is also a pioneer in AI methods for antibody design, having developed RefineGNN and HERN. His recent work includes DSMBind, an unsupervised approach for binding energy prediction, and BindEnergyCraft, a framework for improved protein binder design. We really appreciate him speaking for us today, and whenever you're ready, you can get started.
Yeah, thank you so much. Thank you so much for Divya, Leo, and the organizers for hosting me. It's my pleasure to give my talk today. Thank you everyone for joining today. My talk will be focused on generative AI methods for biomolecular design. I know this is a relatively broad title, but it's because we will talk about not just proteins, but also covering small molecules and RNA design stuff. So, of course, I think this audience more or less know what AI has been, the impact of AI on biomolecular design. There has been a lot of exciting news on AlphaFold and more recently like BOLT, Chai, and all these structure prediction models. And of course, their applications to drug discovery or protein engineering, right? There's RF diffusion and many models that are coming up, right? So during my PhD time, we did use a graph neural network to predict, molecular properties, and which is the ChemProp model, and we successfully used it to discover new antibiotics that are just using pure, simple, virtual screening method. We already discovered a powerful antibiotic, which was recognized by the top biology journal, Cell, right? And since then, I have been working in this biomolecular design field very actively, right? And more recently, there has been a lot of success in protein design using generative models to design, mini proteins, antibodies, many things, right? So there has been a lot of exciting progress in this field as well. So generally, biomolecular design has three components, right? You have the generative model that can generate biomolecules with desired properties, and then you have the scoring model that evaluates whether a designed molecule has the property that you want, right? And then you work with either collaborators or with like some sort of automated lab to experimentally validate these designed molecules. So if you just look at from the machine learning side, how can we formulate this problem, right? So we can sort of map the key of this biomolecular design problem is to build this molecular property relationship, right? You can see from this figure, right? You have these biomolecules mapped to their properties, right? So, and you have a variety of properties, right? You have for example, in drug discovery, we could, in the case of small molecules, we might want to design a molecule that can kill the bacteria, the virus, or cancer cells, right? So then you care about whether this molecule can be, antibacterial, antiviral, anti-cancer, right? And at the same time, you care about their toxicity or their ADMET profiles, right? Whether they can go into the body and behave well in vivo, right? It's in a structure-based, drug design. We care about its binding affinity to proteins, right? And here, in terms of biomolecules, we not only my lab not only just work on small molecules, we also work on protein design and also RNA engineering, and they have very interesting applications in different biological fields. So today my talk will mainly focus on like three sort of vignettes on how we can use generative AI models to design a variety of things, right? So we will talk about three vignettes, one on small molecule design, one on proteins, and one on RNAs, right? And so this is more like a three-course meal for people to kind of just get a, for those who don't know the field yet, you can get a glimpse of what this field looks like. For those who are in the field who are very familiar, there's some of the parts have interesting technical details, technical sort of innovations that you might find useful for your research, right? So the first vignette today is about generative design of novel antibiotics. This is a collaboration between my lab, and Jim Collins' lab at the Broad Institute. And we recently had a new paper that's published in Cell that uses generative AI models for antibiotic design. Yeah. So why do we care about antibiotics? Antibiotic resistance is actually a big problem, right? So if you look at this figure, you have a different kind of disease, and their mortality rate, right? So cancer, of course, right now is the biggest sort of disease. And AMR stands for antimicrobial resistant infections, right? So these are caused by these resistant bacteria, which is also known as superbugs, right? And they are immune to or resistant to existing clinical antibiotics. So the WHO has actually predicted that by the year of 2050, you will reach about it will be actually the biggest death factor in among all the diseases, matching cancer. And we haven't been also what's worse is we haven't been making good progress in finding new sort of antibody classes, right? So this is a sort of a trajectory of antibiotic discovery since the discovery of penicillin, right? So, of course, penicillin is discovered without AI, right? It's about it's a very interesting story, right? And it was discovered around year of 1920. And then we started to, have a sort of a boom of discovering all kind of antibiotics. They mostly categorize into six different uh, sort of categories. But since these low-hanging fruits are kind of discovered, they are mainly discovered through natural products. So basically, you go through different kind of plants, and you found that a lot of plants are immune to bacteria, and you kind of study them and find, oh, there are some chemicals in those plants that are antibacterial, right? So that's how the early stage antibiotic discovery was. But once these low-hanging fruits are kind of picked, you start to actually struggle in finding novel antibiotic classes that are different from existing um, antibiotics. So there are some improvements of antibiotics in these categories that has been approved, but the bacteria are developing resistance to these very quickly. So, of course, antibiotic discovery is hard because of the large search space of the biomolecules, right? So you have, 10 to the, sorry, you have about, this whole chemical space is about like 10 to the 60th, right? And you could, it's like finding needle in a haystack. You can use in industry, you could use these experimental screening to kind of go through them one by one, and then try to, if you are lucky enough, you can find a candidate that decently promising, right? And but even say if you want to just screen through about 1 million compounds, it would already be so costly that you won't like to pay for that, right? And of course, the scalability is also a concern, right? You can't really scale up this kind of experimental screening to the full chemical space, right? You can go through millions, or with, for example, more better technologies. For example, like these combinatorial screening libraries. right? You could potentially go to 10 to the ninth, but it's still so far away from this entire chemical space, right? So ideally, we'd like to, instead of just going through this kind of screening process, going through them one by one, we like to do something a little bit smarter, right? I want to, search for chemicals using sort of generated models, where a smart agent that can generate a sort of molecule that just works, right? So this is like generating molecules. So you could, the general kind of workflow looks like the following, right? You have this generated model that can sample molecules, right? You can sample all kinds of molecular structures, and you can evaluate them, right? Evaluate using a in silico reward model, right? So it's like ChatGPT, where you have this generator and you have this reward model, and you do some sort of RLHF, right, to kind of improve, let the generated model gradually focus on, gradually climb up this hill, right, to find molecules with the best property. So that's kind of the vision of generative molecular design. And of course, after you went through several iterations, you would like to kind of test these molecules experimentally, right? So this is what we are going to do in this part of the talk. So let's go through different. So we have two components here, right? One is the generated model, and one is this property predictor. For this part of talk, we'll stay a little bit high level because some of the work that we have done are relatively known already, and but let's go through the general workflow first, right? Um, so so through the collaboration with people at the Broad Institute, we collected, we did some experimental screening, very small, around 10,000 or 40,000 molecules. We tested, basically we put these molecules into these bacteria and see what kind of molecules will inhibit bacterial growth or essentially just kill them, right? So, and we generated such data, and of course we did in multiple replicates, and we found that our experimental screening has a good agreement, right? So the result is reliable. So we collected about, and then we binarized the output, right? So here you can see the red ones are labeled as inhibitive, right? So these molecules can inhibit the bacteria. The blue ones are the inactive compounds that fail to inhibit the bacterial growth, right? So we generated such data sets that can teach the model to predict what kind of molecules are um, active or antibacterial. We also care about cytotoxicity, right? So we don't want to kill the bacteria to the point that we also kill human, right? So we want to find a molecule that is safe. So here we also generated a similar, using similar screening technology, but for each molecule, we test whether it kills the human cells or not, right? So we actually took three different cell lines, right? There's the liver cell line, there's also a muscle cell line, and there's another cell line as well. So we measured whether when we put these molecules into these in these human cell lines, we measure human cell viability. So we want to select molecules that keeps the human cells viable. So that's how we measure the cytotoxicity of these compounds. So then we use, based on these two data sets, right? We train graph neural networks to predict whether, because these molecules are graph structures, so graph neural network is kind of the ideal candidate such for predicting molecular properties. So we train this graph neural network to predict what kind of molecules will be antibacterial, right? So we basically do this binary classification task where we have molecules labeled as positive and a negative, and we teach the model. We train the model in a tenfold cross validation and generate an ensemble model to predict the antibacterial property of any new molecules. We do the same thing for cytotoxicity prediction, right? Try to predict what kind buildings on top of the same model architecture using this ChemProp software. We predict what kind of molecules might be cytotoxic. And we and we train these two models, and they will be used as the reward function to guide the generated model. So in terms of molecular generation, I know there are actually a lot of molecular generated models these days, and for in this context, we actually used this graph-based generative model called junction tree variational autoencoder that operates on fragments, right? So it's like if you're not familiar with small molecules, you could imagine all these functional groups or these fragments as some sort of Lego pieces, right? And you want to teach the generated model to put these in the correct order so that they are chemically valid, right? And so this JT-VAE model basically generates molecule autoregressively by picking these functional groups one by and stitching them one by one, right? So we kind of let the model just to generate this molecule in the autoregressive process and use that to propose candidate molecules. And then we plug that into this couple that with a reward model and try to find molecules that has the best predicted properties. So that's kind of how we do this molecular property guided molecular generation. Right. So, of course, this concept has been kind of developed a while back, around 2018 to 2020. But this part of the talk is really to show that this kind of methodology works, and we demonstrate the effectiveness of such workflow in the context of antibiotics. So we will look at two case studies on whether this workflow would work, right? And I'm sure there are many other type of approaches, different kind of generated models that can be plugged into this. But I think this work is kind of among the sort of the more serious campaign on validating this kind of workflow in wet lab, right? So the first case study is to find antibiotics for this bacteria called gonorrhea. And it's a resistant bacteria that's labeled by WHO as a urgent threat, right? So it's causing about 1 million sort of infections every year, and about the economic cost, damage to the world, is about 100 million, right? So it's actually a pretty bad bacteria, and we really don't have we'll actually only have one clinical antibiotic left that still works for this bacteria. So it's kind of challenging. So there is a urgent need to discover new antibiotics for this bacteria. So our lab has achieved success in the past in this kind of virtual screening campaign where you just use your graph neural network and just use that to screen through, say, a billion compound library and try to find molecules that work. We tried this virtual screening campaign to our compound library, and we didn't find it to work. I mean, it works to a certain degree, right? So we found two molecules in this chemical libraries that has about 10 MIC minimum inhibitory concentration around 10 to 16, right? So the potency is around that range, which is not super exciting, but also we found that it has a relatively poor selectivity, right? So hold on, sorry. If you look at its activity to human cells, right? If you just double the dosage of these two compounds, you can already kill the human cells very well. So this is not an ideal sort of type of candidate because they have pretty low selectivity, right? Ideally, you want to kill the bacteria with a very low dosage, but having requires very high dosage to be toxic. So you want the difference, large difference between these two, right? So we basically we decided, okay, so that's probably because just looking at this compound library is not enough. Maybe our silver bullet, the best candidate, is probably somewhat de novo. That's not really in this a chemical library, right? So we basically used this generative design campaign, right? And we actually just tested two compounds that because of our sort of budget, we think we generated many candidates, but we eventually went for two compounds and NG1 and NG2, and one of them actually worked, right? So I'm not showing NG2 here, but NG1 worked very well. So this green curve, as you can see on the left, is the dose response curve for the compound NG1, and it has this potency around 0.5 milligram per milliliter, right? And you can see it has around the same uh, sort of toxicity, but you can see the selectivity goes up much better, right? So you can see that this compound improves the selectivity from 2 to 64 compared to everything compared to the compounds that initial hits that we found in BRD1, BRD2, right? So the black curve and the red curve are the dose response curve for two different human cell lines. And you can see there's a good sort of separation here. This is exactly what we want. Of course, this is an in vitro result, right? So we'd like to see how well this compound does in vivo, right? So we did this in vivo study in mice. We tested these compounds in mice models, and basically we divided like about 18 mice into vehicle is the control group where it's basically placebo, it didn't receive anything. And we have this compound NG1 treatment for another six mice, and we have a positive control. This is the clinical antibiotic that still works. It's the only drug that still works for this bacteria, which has the kind of the last defense line of our battle against gonorrhea. So you can see that from this figure on the right, you can see this is the y-axis is the infection level, so it's the lower the better, and you can see the vehicle control, and you can see this middle column is our compound NG1, and of course the clinical antibiotic works very well still. But you can see our compound is fairly active in vitro as well, right? It can reduce the infection about 100-fold, right? So, and you can also see that our compound not only kills the wild type gonorrhea, but also kills different resistant bacteria strains. So here's another figure of kind of multiple different gonorrhea strains. You have the wild type, which is on the first column, and the second column is sort of the resistant strains that's collected by CDC. There's also another two even worse strains that's pan-resistant, meaning is resistant to this compound as well. So it's kind of very important to have something that works for these two resistant bacteria strains. So these results shows that our compound actually is quite useful in vitro, right? So this is the first case study for this bacteria called gonorrhea. We also looked at another bacteria strain called MRSA. This is relatively more well known. It has if you buy Clorox, you will see MRSA in the sort of the description that they claim that they can kill MRSA, but of course it's because it's like a soap, it can kill everything, right? So it doesn't have much selectivity. So we do want to find compounds that are selective against MRSA and try to kill these bacteria, right? So here we also did a similar campaign, right? We trained the model using a different data set collected for MRSA, actually for for this bacteria, and we generated and tested the top 24 compounds selected from this generative model, and we found these are the compound structure of these six designed compounds, right? And you can see that the top one compound, DN1, actually achieved potency around a single digit potency, but of course the selectivity is good, right? So selectivity is about 64 again. So this compound is it is safe and it's effective against this bacteria, MRSA, right? So we similarly tested this compound both in vitro and in vivo, right? So we again put these compound in mice model, and you can see that for DN1, we can reduce the infection about tenfold. It's not surprising. It's not like a home run, but it's still active in vivo, right? And you can see on the left-hand side, you can see this compound actually kills bacteria faster than a clinical antibiotic for MRSA, which is called vancomycin, right? And you can see this is the blue or the purple curve is the curves for dose response curve for or time. This is a time killing assay. So you basically look at if you put compound into mice and see how long does it take to kill the bacteria, and you can see that our compound actually kills bacteria much faster with the same dosage as my vancomycin. So in general, these two results kind of show that this generative modeling um, campaign actually is quite effective, and we actually demonstrate this the effectiveness of this method in vitro and in vivo, right, in animal models, and we hope to translate these compounds into preclinical studies for to further test it on eventually on humans, right? So these are kind of our summary of the first part of the talk. So of course we this the sort of the small molecule is not the only um, type of drug, right? So there are proteins, biologics that are also useful for different kind of disease areas, right? For example, in cancer immunotherapy, a lot of these antibodies or mini proteins that are effective as these checkpoint therapies, right? That can reboot our immune system to fight against cancer, right? So we are equally interested in other kind of biomolecules. So in the second part of today's talk, we'll talk about how we can design proteins effectively that binds to different kind of targets, right? So here the property that we care about is the binding affinity to proteins, right? So yeah, and of course yeah, so there has been recent success in protein binder design, right? You have different two. There are mainly two types of methods, and I'm not saying there's everything falls into both to these categories, but of course there's protein language models as well. But if we just look at the kind of the most well-known type of models these days, one is diffusion based, like RF diffusion that can generate protein binders. There is also a hallucination based method called BindCraft, developed by groups at EPFL. They can design binders in one shot just using like 96 well plate. You can actually discover some binders with 100 nanomolar activity to like single digit nanomolar, which is very exciting, right? So and that can save us a lot of experimental effort, right? So it's a very exciting field. So we dived into this is actually work done by Divya, and we went into this BindCraft model and we tried to play with it, and we found that this method, of course, so the at high level, BindCraft works as follows, right? So you have this AlphaFold 2. It can be AlphaFold 3 as well, but the current version is AlphaFold 2, right? And you can feed your target sequence and its structure along with the binder sequence. You feed that into the AlphaFold 2 model, pass through its evoformer, and you try to predict the structure and predict the confidence of the predicted structure, right? And these confidence scores include like IPTM, PAE, or PRDDT, etc., right? So, and the basic idea of BindCraft, for those who don't know, is to basically iteratively update your binder sequence to improve these confidence score. And the rationale behind this approach is people have found that these confidence scores like IPTM are quite predictive in binding. So they can predict protein-protein interaction or distinguish the binders from non-binders with a decent level of accuracy, right? So that's kind of what we are trying to. So that's the reason why BindCraft proposed to basically do this gradient-based optimization, try to backpropagate through AlphaFold, right, to optimize the binder sequence to improve a combination of these um, in silico binding like confidence scores. But when we investigated this in more detail, we found that some of these objectives are not as smooth as we expected, right? So, for example, this IPTM, which is one of the important indicator of binding, uh, its gradient is not smooth, right? So because IPTM actually involves a max operation over all the residue. So when you backpropagate IPTM towards the binder sequence, you actually only one residue gets updated, right? So because it's the one that's selected by the max operation, right? And you can see that this kind of sparse gradient intuitively will make our optimization hard, right? So ideally, we like to find something that's smooth, right? We would like to find something that can easily optimize, but also predictive of binding, right? Um, so we would like to find something that kind of work like free energy score, right? This is kind of going back to physics, tries to learn this kind of smooth energy landscape of different conformations and their energy score, right? And to find different sequences um, that has low energy. So that's kind of the motivation behind our sort of design method, right? So and another reason that we wanted to so the focus of this part is try to curate a better, smoother sort of in silico objective for binder design, right? So, and our motivation is also sort of inspired by recent uh, 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 methods on like language models, right? So, and for example, if you are if you're familiar with language model in natural language, you actually know that these language models are trained to optimize the likelihood of these natural sequences, natural sentences that collected on the web, right? And if you have a sentence that has high likelihood under a language model, then you're probably confident that this sentence is grammatical and makes sense, right? And similarly, in protein sequences, if you have people have trained protein language models, and you can see that if you have a good likelihood of the protein sequence, then it means if that sequence has a pretty high likelihood, it's highly likely that this protein has good fitness, right? Driven by these sort of ideas from sort of related fields, we hypothesized that maybe for this binding, right, we should find a way to assess the likelihood of the protein structure or the structure of the protein-protein complexes, right? And maybe if a binder target complex has a high uh, likelihood, then it might be that they will highly likely to bind, right? So that's kind of the basic idea behind our model. Like we try to find a in silico score that actually is driven by the likelihood. So that's kind of the motivation behind our work called PTM energy and BindEnergyCraft. So the rest of the this part of the talk is try to come up with a way that can learn or extract this energy efficiently, right? So here the approach that we took is we actually found a way to cast AlphaFold as a energy-based model and to extract the unnormalized likelihood of these predicted protein structures, um, actually based on the confidence heads. So the basic idea behind our this PTM energy idea is motivated by a relatively early work done by David Dumano's group in University of Toronto, and they had this paper called "Your Classifier is Secretly an Energy-Based Model." and their method is called JEM. So this JEM framework identify that if you have a classifier, right, say you have a image and you put that into some sort of neural network and try to, based on after that you predict the class probability, right? Well, whether this cat is whether this image has a cat or dog or something else, right? And of course, this kind of classifier itself doesn't really give you the likelihood of that input image. But what this JEM framework discovered is actually, there's a way to extract the likelihood of a image of the input from a discriminative classifier. And the basic idea is to look at the softmax operation, right? So this py given x is if you write out the softmax operation, right? Y stands for a different class, and x is the input. In this case, can be image, but in our case, could be a protein, right? And based on this JEM or joint energy-based modeling framework, you could actually reuse the logits. Logit meaning the fxy. You can reuse these logits to define a joint distribution px comma y. And of course, now you're just normalizing over, you have this normalizing constant z, but it's no longer normalized over y, but normalized over x, right? So, and take the, if you marginalize the y's out, right? If you just take the sum over all the y's and take the log of it, then you have log px and equals to log sum exp of all the logits, right? So, and you can see this is basically the normalizing constant of this softmax operation, plus some constant related to this z, right? Which is a constant. And so you can see that if we just calculate this log sum exp from the or take the basically the take if you take the normalizing constant of the softmax, you actually get this likelihood of an input image, right? In this case, even though this model is trained to do a classification task rather than a generative modeling task. So why are we talking about this, right? So if you look at AlphaFold more carefully, you can find that these confidence heads are actually a classifier, right? So you predict the PAE logits, right? So you have this predicted structure from AlphaFold or from Boltz or from F3, whatever, right? You put that into PAE head and you actually take the softmax and to predict this PAE logits, right? Basically, which bin will it fall into, right? So we can apply this idea to this PAE classification head, right? Now we can take the energy of calculate the unnormalized likelihood, or AKA energy of a statistical energy of a predicted complex, and calculate their statistical energy using this joint energy modeling framework. And this doesn't require you to fine tune or any part of AlphaFold, right? And you can get this. So basically, what you need to do is instead of going through softmax and aggregate these class probabilities, you don't do softmax, but you take the log sum exp of all the logits, and you can actually get a log p structure, right? The structure of the predicted complex, and you can use that to calculate use that as a way to rank different designs design proteins, right? So, and of course not every PAE being is equal. Higher PAE being means worse binding, so we actually apply the TM weighting to different PAE beings, and this gives us a PTM energy, which is similar to this IPTM score, but it actually gives you as I will show later, gives you a smoother landscape. So let's first look at whether this PTM energy is how well does it do in distinguishing binders from non-binders, right? So, and here is basically we took about 14 mini protein binder screens from two papers published in David Baker's lab. They conducted about 14 different binder screens that they designed using either their oldest Rosetta like physics-based models or more recent Rosetta RF diffusion models. And they basically have some are binders, some are non-binders. And we used we basically for each target, right? so there are like 14 different targets. For each target, we downsample the data set into 1 versus 100 positive negative ratio, and we calculate their IPTM or PTM energy score to see whether what kind of in silico score is predictive that can distinguish binders from non-binders. And you can see from on this table, this AUPRC of our model of PTM energy actually gives better, higher, it actually performs better than IPTM. We also tried a smooth version of IPTM by replacing the max operation with a mean, but we still found that our method performs better in terms of AUPRC and also the precision at 5 at 10, meaning the hit rate, if you just take the top 10 and if you just select those and go for designs, right? You can see that our hit rate is actually higher. So yeah, so now you can also look at the PTM energy score distribution across, there are 8 different targets, and you can see here are the red bars are the positive true binders identified experimentally, and you can see that for majority of the targets, our model can rank uh, binders at the top, right? And this is the x-axis is the negative PTM energy because our score is actually the lower the better. So we just to be intuitive, we flip the sign, and so here should be the higher the better, right? So and you can see for one of maybe this H3 wasn't doing that great, but others actually you can put the binders at fairly at the top, right? And we actually tried this on looked at whether this can be useful for RNA design as well. And which we'll kind of talk about that a little bit later. We can use that to predict the binding. So here we took a GFP aptamer data set identified by a Celix screen. From this 2012 paper, they basically have a bunch of RNAs that can bind to this GFP, and you have a bunch of others that can't. And you can see on top of Rosetta fold NA, if we plug in this PTM energy idea, you can see that actually we can get a 10% improvement in terms of AUPRC and also the precision at 10 at 50. Right, so this shows that this PTM energy is actually a good binding indicator compared to IPTM and others. So now coming back to our initial sort of motivation, right? We were trying to find something that is uh, driven that has solid sort of probabilistic interpretation, and also try to find something that is smooth, right? So this PTM energy actually is as a smooth metric, right? So because it's a log sum EXP, if you differentiate this log sum EXP with respect to the input sequence, you actually see that this is the heat map that you can see that this is well spread across all the residues. So when you do one step update, you actually it's passing a lot of information and you can actually get a lot of updates. So you can actually do optimization much faster. That's kind of the basic idea behind why we're trying to switch to. So basically, what we do is exactly the same as BindCraft, but we replace this IPTM with PTM energy, and this gives a smooth gradient. And even just with that small like change, and you can see so we basically did plug this in. We compared BindEnergyCraft with the standard BindCraft method on 8 different targets. And basically we use the same in silico filters, right? So even though we're optimizing PTM energy, just to be fair, to compare with all the existing methods, we still use IPTM as one of the sort of the in silico metrics to define whether this binder passed the in silico filters, right? So, and of course, these are the filters we copied from BindCraft, which they found to be very useful for prioritizing their designs, right? So you can see that with our BindEnergyCraft, you can get around like 5 to 10% improvement compared to BindCraft. And this is not only because our model is PTM energy is smooth. Even so, we tried also a smooth version of IPTM with this mean pooling instead of max pooling, which supposedly is smooth, but you can still see that our model, driven by this energy score is actually it actually does better. And this kind of shows that this probabilistic interpretation based on the likelihood is actually a better way of predicting binders from non-binders. We also found that this BindEnergyCraft operating on a better energy-based score actually reduces the clashes between the designs and between the binder and the target. So here you can see comparing to BindCraft, you can see a much less number of clashes across 8 different targets over the board. So this kind of shows that driven by this statistical energy, it can actually make generative to we can it actually helps the model to generate better sort of more biophysically feasible designs. So that kind of shows that this is a very interesting twist of AlphaFold, try to twist that into an energy-based model that resonates with the old days, like Rosetta or these methods that try to design proteins based on energies. And this is kind of falling into that category, but not purely physics, right? But has that kind of flavor. So this is kind of interesting, right? So this is that kind of concludes the second vignette on the BindEnergyCraft and how we can use that to design protein binders. So in the last part of the talk, we'd like to go through um, move to RNAs, and here we're interested in a type of RNA called aptamers, and which can bind to small molecules or proteins, and they can be quite useful. So RNAs is something that's getting attraction these days in these ML conferences. People are starting to look into RNA design as well, right? So RNAs can be engineered to have versatile functions. So like aptamers are also known as like equivalent form of antibodies, but in terms of nucleic acids, right? So they can be useful for therapeutics as well. But of course, there's also other kind of RNA like riboswitches that can control gene expression that can bind to different like transcription factor domains, and you have ribozymes that can modulate protein activity. So RNA design is actually a very useful, interesting field as well compared to proteins, right? So of course, current RNA design approaches, a lot of the traditional approaches are driven by high throughput screening, and this is one of the most well-known screening technology is called CELEX, right? And you basically go through this kind of in vitro selection evolution that kind of you have a pool of RNAs and try to do some error-prone PCR, tries to induce some mutations that let they evolve, and you kind of try to screen and try to enrich those that are functional, that sort of thing, right? So of course, this is very costly and time-consuming and laborious. Right? So we'd like to go to de novo RNA design, right? So this is done before BindEnergyCraft. So BindEnergyCraft, if we currently operating on R42 doesn't have that capability. But before that, Divya and I, we developed this model that's called RNA flow that's sort of motivated by the success of RF diffusion in the field of protein structure generation, right? So you have this Rosetta flow that can propose new structures, and you try to do this kind of diffusion model to generate structures iteratively, right? So we wanted to propose something similar for RNAs, right? Try to generate these RNA structures using some sort of folding models, like our Rosetta fold NA, for example, right? But what we found is actually fine-tuning these. So like RF diffusion had to is fine-tuned from this Rosetta fold, which is a big sort of beast that has a lot of parameters. And if you just fine-tune these large structure prediction models, it's quite computationally expensive, right? And it runs very slow. It optimizes very slowly. So we kind of start to ask ourselves, is there a way to kind of do it in a more efficient way? Can we generate, and also this is not that feasible for RNAs because RNA crystal structure we don't have that many crystal structures for RNAs compared to proteins, right? So we want to have something that's more sort of cost effective and also being efficient, right? So this is how we came to develop this idea of RNA flow, which is use a flow matching model for RNA design. But the key contribution here is that we want to generate structures without expensive fine tuning of these expensive protein folding models. So this is the sort of the key twist of RNA flow. But basic idea is, and here we want to design RNAs that binds to proteins, and which is important for RNA aptamers. So here is a sort of so basically we have this protein backbone and noise RNA complex as an input, and we want to feed that through RNA flow and try to predict the sequence and to generate the sequence and the structure um, of the RNA that presumably binds to this protein, right? So, but the key contribution here is we want to do something that's not fine-tuning Rosetta Fold NA or any of these expensive neural network. So we actually took inspiration on the fact that RNA, RF2NA, all these protein structure predictors already learned a good sequence structure relationship, right? So you don't really have to if you want to generate structures, you can actually try to find a way to steer these existing protein folding models to generate things that we want, right? So, and the denoising structure, we don't have to repurpose R2NA as a denoiser in the diffusion process, right? We could actually train a separate module that try to propose sequences and use this sequence generator to as a denoiser to generate the structures. So basically, the idea is if you have a sequence structure relation, if you have a model that can map a sequence to a good structure, then you just have to do the sequence design part in order to generate good structures, right? So that's kind of the basic idea behind RNA flow. Uh, so basically, we will train this inverse folding model as a denoiser that can, given a proposed structure, try to infer what kind of sequence will fold into that RNA structure and feed that back into RF2NA to update the structure. So, but the RF2NA part is fixed. So let's go through this step-by-step illustration of how RNA flow works. And it's relatively intuitive. So you have this sampled prior, right, from a Gaussian noise. You have this RNA that's purely just noise. So what you do, right, you just feed that into this inverse folding model, try to propose some sort of sequence, right? And the sequence can be quite arbitrary because the structure, sorry, this is a purely just random point cloud, so you can't really get a meaningful sequence out of it. But nevertheless, right, just let it propose something, and now we reconstruct the structure from this sequence, and you get something, right? So now you actually update it, that noise, right, through this two-step process, right? The inverse folding model is trainable, and RF2NA is frozen, right? You kind of mapped that into, you kind of map that into some sort of meaningful RNA structure, right? And of course, if you want to do this, you have to do this iteratively, right? To flow matching, right? So what you do is you actually linearly you re-noise it, right? This is similar to standard flow matching or diffusion models where you always try to predict the x0, right? The ground truth, and then you add noise back. So you can add the noise back to add this noise on top of the predicted structure R0, and you get this R1 here, right? So this is a linear interpolation between the initial sample and the predicted denoise structure, right? Now, what you do is you go through this inverse process, a folding process again, right? You get this inverse folding model. But now, since your structure looks more decent, right? You actually get more information out of it. Now you and then you feed it back to RF2NA, and you get some sort of a new sort of structure again, right? And you just and you re-noise it, and you kind of go through this process iteratively, right? So you can actually now what you can see here is you just need a cheap inverse folding model, and you can actually do this flow matching, you know, prediction, right? You don't need to fine tune RF2NA, right? And of course, the loss function is defined over, still defined over, similar to RF diffusion, is still defined over the structures, right? So you always want to, so you just minimize the mean square error between the velocity of the from the model to the ground truth velocity that points to the ground truth, right? So you just do that, the standard flow matching, but you only train the inverse folding model, right? So you just update this inverse folding model, which is relatively cheap, and the Rosetta flow to NA is frozen. So that's give us a much better, much lower number of parameters and much more scalability, right? Yeah, and of course, we could also add, in these RNA inverse folding models, like gRNA-DE, they actually have a ways to condition on ensemble of structures, right? Because RNA structures are flexible, so you could actually, instead of just conditioning on your immediate uh, current kind of noise structure, you could actually condition your inverse folding model on a trajectory of this process of the RNA structures, right? So you can actually feed that into a multi-GNN encoder and do some sort and decode the sequence on top of the structure ensembles. So this is following sort of the multi-state RNA inverse folding methods developed recently, right? But we apply that to RNA design, right? So that's kind of the high-level idea of the method. So we evaluated our model. We compared to pure sequence-based models and some diffusion-based models for nucleic acid design that people have developed previously. And we evaluated two variants of our model. One is RNA flow-based, which is just conditioning on single structure, but we also had a RNA flow trajectory that does inverse folding over the trajectory of that ensemble. So we here we look at basically selected a few RNA structures as the test set, and we did sequence similarity split. We actually did two types of split. One is the temporal split, one is the sequence similarity split. And we trained the model on the PDB with these test set hold out, and we evaluated whether we can actually recover the sequence on these on the test set. And you can see that our model with RNA flow trajectory, we have some additional twists like try to rescore, add some rescoring modules, which I will not go into details due to time. We can see that you can improve the sequence recovery rate to up to 37% if you do RNA flow trajectory. And you can also see that the predicted RNA structure also agrees with the ground truth with a good LDDT score, right? So, and you can see that with if you just feed the ground truth sequence to RF2NA, you can get around 0.8 LDDT. But if you use the design sequence, right, and you feed that into the to RF2NA, you can see that it has a relatively similar structure to the ground truth that bind the ground truth RNA structure that binds to the target. So that kind of shows that without this expensive fine tuning, we can actually just adding some sort of adapter, we can actually get a fairly good RNA sequence structure co-design model, right? So these are some of the examples of degenerated sequences. And you can see that in some cases, RNA flow actually predicts the cross structure using generates some generates a structure that looks similar to the ground truth, but with a totally different sequence. So that's kind of quite interesting. So we also plugged into a one case study. We tried to see whether we can design an aptamer that binds to this target called GRK2. GRK2 is a target of interest for chronic heart failure treatment. And there is a crystal structure of, people have found through experimental screening, people have found a RNA aptamers that binds to the target strongly. And people found that there is a 4 nucleotide motif that is critical for the binding. If you change any of these motifs, you will actually lose binding. So this gives us a way to test, instead of just looking at the sequence recovery, we can look at whether we can generate sequences that has that motif, right? Because when we have that motif, it's known that they will very likely to bind. So this actually gives us a good way of evaluating um, whether the model works. And you can see that this RNA flow trajectory that can gives us a recovery. So here the recovery is the how many of those design sequences recovered that has that 4 nucleotide motif, right? So you can see that we actually have about 54% of the design sequences that contain that critical binding motif, without telling the model that you know that this has the motif, right? So this is kind of the idea behind RNA flow, and we show that some of the design structures that closely aligns to the ground truth, right? So these are some of the visualization of the predicted or designed RNA optimal structures with GRK2. Okay, so that kind of, I know that I went a little bit over time, but here is a summary. Yeah, I shared 3 vignettes about how we can de novo design small molecules, proteins, or RNAs, and we work with experimental collaborators on some of those already to validate whether these are actually working, and we are actively pursuing collaboration on the protein and RNA front as well. So here we just want to acknowledge, the agencies that funded our projects, but also the wet lab collaborators for antibiotics in Jim Collins' lab that really helped us. And also want to thank Divya, Anisha and others for the development of different models. So I also put the links to BindEnergyCraft and RNA flow. Our antibiotic design paper will be featured on CSAIL very soon. So yeah, thank you so much for listening. And let's see if you have any questions. I'm happy to go through some informal discussions here.
Thank you so much, Wengong, for an amazing talk. Yeah, feel free to leave questions in the chat. We have a few already. I can read them out and we can go through. So someone asked, are the potency and selectivity values determined from experiments or predicted by an ML model as well?
Yeah, thanks. That's a great question. So the potency and selectivity values are determined from the experiments, right? So if I go back to, let's see if I can move back to here, right? So if the selectivity here is not predicted by the model, but we did an in vitro like assay to test whether this compound can inhibit this bacteria, and we did so. These selectivity are all done in vitro in bacteria and human cells. Hope that answers the question. Of course, the design during the generation process is driven by the predicted models. So we basically try to generate compounds that has high likelihood of killing the bacteria, but low score of cytotoxicity.
The next question, what do you mean by statistical energy? How do you calculate it for a predicted protein structure from AlphaFold?
Yeah, that's a great question. So the statistical energy is basically the PTM energy score that we calculated here exactly. So on top of the pre-trained uh, like the confidence score, right? We can actually this PAE head, we can actually calculate instead of calculating IPTM, we can calculate this PTM energy. That I already explained why it is energy score. So this is what I mean by statistical energy. So the reason why I use the term statistical is because this is not a physics-based method, even though it has the term energy in it. And statistical energy, if you are familiar with energy-based models, right? These um, uh you can yeah, like it's the energy score, if you normalize it you could get the likelihood. So the unnormalized likelihood is also known as energy. um yeah, so that's why I think I kind of use the term statistical energy.
How is AlphaFold 2 PTM energy compared to AlphaFold 2 IPTM on the mini protein binder prediction task?
Yeah, that's a great question. I think I probably showed this or in this part. I think it's the both one. We didn't show that in AF2. So I think we do have to look into that. Whether AF2 multimer, how does that PTM energy compare to the IPTM? But at least in terms of both, we show that it actually does pretty good.
In slide 15 for the JT-VAE generative model, is the input to the model the set of desired properties? And if this is true, then are these properties the same as the ones we've used for the property prediction models?
The answer, yeah, yes. Like we are basically looking at two properties. One is the predicted potency, the other is predicted cytotoxicity. And we also, after we finish all the generation, we do have some additional filter on the some of those are based on the synthetic accessibility, which we could have added during the generation, but we didn't. That was a mistake, but we already proposed the compounds, and we found that two of them could be synthesized by enamine. So we just went ahead and we just actually came back. One actually was good. So, but yeah, I think so. The first part, I will say the method is still a little bit kind of shaky, that I think there's a lot of room for improvement. But we managed to just in a short time frame actually did this and show that it actually works.
On
Oh
slide
yeah.
29,
So
sorry.
yep.
So we
Yeah,
have
go
a
ahead.
couple more questions. Thank you for your
Go
answers
ahead.
so
Yeah.
far. In slide 29, PTM energy, what hindered you from making a dedicated binding affinity predictor and maybe use it as a reward or feedback instead of the likelihood as an estimator?
Yeah, that's a great question. I think right now, like for example, we tried the supervised model, just train on. I know there can be so like if we have enough protein-protein interaction data, we could use that and use that to guide the generative design. That I think is a good area of exploration. I think there is people should look into this more seriously. I am interested in that direction. So far, what we found is if you just train so this supervised model is a fine-tuned version of sorry, like a building on top of a ESM2 language model and try to. So basically, there are like eight. Oh, I forgot it's 8 or 14 different mini protein binder data sets, right? So we basically did a cross-target cross-validation. We say we train on a subset of targets and see if the model trained on top of these targets can generalize on the other set of targets, right? So um, and you can see that these supervised model actually don't generalize well across targets. And this sort of unsupervised sort of the likelihood or IPTM score actually generalize better across the targets, which is surprising. So maybe we could think about some other ways of merging, different experimental data with these IPTM or likelihood-based scores and to come up with a better metric. These are all like open areas of sort of exploration. I'm here to propose ideas, and I think there's still a lot of room for different ways to improve on top of this.
In slide 42 for RNA flow, was there any specific reason for using flow matching for this problem specifically?
Great point. Great point. I think just in general, we found that flow matching is just much easier to train, because the flow is straight and it makes the generation process simpler. And I think um, for David Baker's lab and a variety of groups, they are trying to move towards flow matching. So I think that is a good sort of, it's just a general rule of a kind of trend back then. And I think, yeah, we could have tried diffusion. We just didn't do that. But I certainly value the possibility that, diffusion would work well here.
Someone asked if I'm reading the plot correctly, NG1 is still not as good as ceftriaxone.
Oh, that's correct. Yeah, in terms of you are absolutely right. We didn't manage to beat the only clinical antibiotic that's still active, but we want here the goal is we want to find something that's novel that because these bacteria are if you keep using um one antibiotic, the bacteria will just eventually, crack it. So we need a new sort of candidates that are novel, that is different from existing antibiotics, and that as long as it works in a decent range, it's already a good progress. And certainly, we are trying to optimize this compound even further to chase ceftriaxone, right? So, but totally valid. Yeah, we didn't manage to get a full home run here. I think there's still like a tiny little gap here.
Okay, we have a couple more questions. How do you verify synthesizability for JT-VAE generated compounds?
Yeah, that's a great question. I think what we did was we collaborated with Connor Corley's lab and used their retrosynthesis tool to measure, what's the number of steps that required to synthesize a molecule and use that as a way to, rank different compounds. But we actually found that when we actually after we screened using that strategy and when we gave to enamine, we found that they actually have a different notion of synthesizability. So there's because of that mismatch, a lot of the compounds that we initially proposed didn't really uh, pass enamines internal their own like human experts. So we only managed to synthesize two. So I think in general this is a still open question of how you actually gauge align with human on like how to synthesize stuff. So for now, we actually used the RA score developed by AstraZeneca. This is open source. You can just go through GitHub, search RA score. This is one score, and the other is the ritual synthesis tool developed by Connor Corley. So that's what we did.
Okay, any thoughts on the pros and cons of JT-VAE versus a discrete diffusion model operating on molecular quote unquote Legos like tokenized functional groups?
That's a great question. I think I just didn't have a chance to really develop such models, but if we can do some sort of discrete diffusion models to this, I think that would be very interesting. Tokenized functional group and learn how to stitch them somehow. I think that's a fairly interesting direction that I will personally be interested kind of to work on that. Of course, I think the current field is moving towards less mouse, which is easier to train. But I'm still remain curious about whether this graph-based method is uh, remain like like maybe the discrete diffusion can actually work better. I totally agree. Yeah, I think I'm very interested in this direction.
Okay, thanks everyone for all the questions. I think we've gone 15 minutes, so I want to make sure we don't take up too much of Wengong's time. Um, Thank you again for an amazing talk and for all the audience engagement, and look forward to the next seminar.
Yeah, thank you so much. Thank you everyone for coming to my talk. Really appreciate that. All right, thanks to organizers too. Thank you so much. Okay.